Hi guys, it's me, Miss Cherry, back again for Module 7. This is Lesson 2, Monohybrid Inheritance. To begin with, as per usual, could you please have a go at answering these questions? You can pause now, spend a few minutes looking at them, thinking over what you know, thinking how it relates to last lesson and your GCSEs, and hopefully this should be a nice, easy reintroduction to Punnett squares. So pause now and play again when you're ready to go through the answers. So the picture itself shows a Punnett square, and that shows the relationship between parental and offspring pheno and genotypes. Between parental and offspring phenotypes and genotypes. And what we can see here is patterns of inheritance. Now we should be able to see, or you should be able to identify, that this true breeding yellow pod is homozygous recessive, using what we recapped last lesson, and that the green pod is homozygous dominant. I am a bit of a lazy writer, so homodom is fine for me right now. Now, how can we describe what is happening? We can use those two key terms, the homozygous recessive and the homozygous dominant, to describe the offspring as 100% heterozygous. And in being 100% heterozygous, that means that all of the offspring have the same genotype, so we can write that in, big G, little g, and they are all green. Now hopefully that has covered all of the questions. If you've still got any more issues with going over this, just pop up on the chat on the call and ask a question. If not, let's get straight into it. As we've said, this is lesson 7.2, monohybrid inheritance. Mono meaning one, and in this instance we are looking specifically at one gene. Okay, so we're looking in that primary example about the inheritance of the green gene, for example. Something like that. This differs to next lesson where we're going to be doing something called dihybrid inheritance. But for now, we're just looking at that one gene. So, a while ago, around the 19th century, there was a monk called Gregor Mendel, and he loved gardening. Okay, specifically, he was interested in looking at peas. And he had a look at what would happen if he bred lots of different types of peas together. So you can see in this top row, we have got the P for parents. And these are the two different phenotypes that he crossed, so purple and white flowers. And he found that in the offspring, or the familial generation, so that's the offspring. F1 is the first offspring. And then if you've got an F1 and an F1, so two offspring breeding together, that gives you the F2 generation. This will be important for later in the lesson, so we'll go over it then again. And he basically saw that there were specific laws of inheritance that these peas followed. Now, in terms of keywords, you should have your Module 7 keywords that I've uploaded to OneNote, or that you will have had access to through teachers if it's not me. Monogenic inheritance is the passing on of a single gene. Monogenic genes from the parent, that's the P, to the offspring, the F1 generation. Now notice we're not writing Formula 1. It doesn't seem to come up with the subscript on here for me, but it should be F and then a little one. Okay, not big F, big 1. Not about that life. Now the way that we can see this offspring inheritance is by using a genetic cross or a Punnett square, and you will have done these very simple ones at GCSE, but unfortunately for A-level there's a very specific set of rules that we have to follow. So let's get started. The first thing you need to do in any Punnett square is to state the phenotype of both parents, what they look like. And hopefully it will say this in the question. Okay? It might also say that G is dominant, or big G is dominant to little g, and big G gives a green phenotype. It might also say in the stem of a question, because sometimes it won't just be very obvious from it, it might say that one parent is homozygous dominant and the other is homozygous recessive. Okay, so this is really important that you're reading the question, looking at the stem, getting all of the information that you can from the question. Okay, so first thing, state the phenotype of what both parents look like. Now, you don't have to draw the pictures. I don't know what this is, maybe a pea flower? I don't know. But what do they look like? Start there. The next thing you've got to do is use the information that you're given, if you're given any. So we said on the previous slide, you need to make a note of the information, check you're reading the stem properly. Then you are going to, after you've written their phenotype, write down what genes the parents have, their genotypes. Okay, now, there's a few ways that you can do this. Sometimes in the question they will give you a key, such as I've got on this picture. But if there isn't, you are allowed to make one. Okay, you can make a key. Now for this, you've got to look at the gene that you're looking at. You can see in this case we're looking at greenness. So they've chosen to use the letter G. You could have also chosen to use the letter G. But there are a few rules. You need to make sure that you're writing capitals for dominant genes, or dominant alleles, sorry, and lowercase for recessive alleles. Okay, this is important. It shows that you understand what's going on and you're not just sort of blindly running your way through step by steps. The second rule to remember when you're making your own key is to make sure that you pick a letter that is slightly related to the characteristics. So maybe you want to use G's because we're looking at greenness. If we were looking at eye colour, you might want to use E's, for example. But make sure you pick one that looks obviously different in upper and lower case. Okay, the problem with some people's handwriting is that if they were to do, I don't know, mottled fur, okay, maybe they're going to do mottled fur and big M is mottled and little m isn't mottled. Both of those M's, when you're in a stressful exam situation, will probably end up looking like that. Okay, and that doesn't show that you know what you're talking about. Okay, so make sure you're very explicitly picking a letter that does look different in capital and lowercase. So a couple of examples down here, you've got A's, you've got B's, don't pick C. You could pick D, E, fine, F, maybe not. Think about your handwriting and think about what's going to look best for you. But please, 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 never, 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 never use X or Y. Because remember, these are for gametes. We're going to be looking at something in a few weeks called sex linkage, and we're going to need to be using those X's and Y's, and those are for the gametes. Okay, so step 2, you're going to state the genotypes for both parents underneath their phenotypes. Now you've got their genotypes, so big G for the green pods and little g for the yellow pods. Notice here that they've randomly assumed, again because they're flowers, and unless you're going to go and have a look, I don't think you really need to worry about it, which ones are male and female. You need to be careful. In an exam question, it might say that the maternal parent, or the mum, has got homozygous genotype, a homozygous recessive genotype, for example, and the dad is a heterozygous genotype. So make sure you're reading that information. Now, through meiosis, this parental genotype is going to be split into gametes. So this is our diploid genotype. Okay, this is what we see in the parent. And we know that undergoing meiosis, thinking back to module 4, we are going to end up with 2 haploid gametes. So what we do is we take that parental genotype and we effectively split it in half. Okay, so you can see down here that my 2, what are they, plant sperm, pollen, from the green pod are going to be a big G by itself and a big G by itself. Okay, and likewise for the yellow pods, if we're talking about the ovary in the plant, we're going to get 2 ovary cells with little g's in them. Okay, now that we've put these in circles, they are distinct things. Okay, we don't stick those 2 big G's together again until we look at the offspring. Remember, these are haploid gametes now. They only need 1 copy of the gene. That's why they're single letters. And now we just do exactly what you've done since year 11, maybe year 10 if you did it earlier. We just use a Punnett square. Okay, so as you can see here, I've decided to put the 2 paternal or father genotypes at the top and the 2 maternal or mother's genotypes, gametes genotypes down the side, and we just cross them. Okay, so we've got a big G here and a little g here, so in the box where they meet, big G, little g, again, big G, little g, and you just carry on filling out your table as you will have done at year 11. Make sure, please, that you're labelling the side of your Punnett square with the gender of each parent if it's known, just so you're showing that you're linking where you've put your gametes to where you're putting them in your table. And finally, the thing that you will have done at GCSE absolutely fantastically is that you are putting the proportion of genotypes produced. Okay, so in this case, we will have put all plants have green pods. You will have put that they're big G, little g. You could have also written 100% big G, little g. You could have written 4 out of 4 big G, little g. And then you need to write down the phenotype that is produced from each of those genotypes. So they've done that nice and succinctly in this little statement here. But if I were to write 100% big G, little g, I could then write underneath it 100% green pods. And that's it. That's it done. Okay, very simple, very easy to follow from your GCSE knowledge. Hopefully we should find the next slide nice and simple to do. So now, using those rules and the example that we started with here, because on the left-hand side this is the one that we've just been over, could you please produce a genetic cross showing the resultant offspring when 2 of the F1 population are bred? So that's a very wordy way of saying what happens if 2 of the offspring have a baby. And to do that, you need to follow all of the rules. There are 6 marks available. Look at the example on the slide and pause now. When you're ready, we're going to go through the answers. So pause. And we're back in the room. Now, marks-wise, it's very simple to gain these marks, but also very, very easy to lose them. So we'll start at the top. We know that phenotypes-wise, we are starting with 2 green pods. Okay, we said that at the end of the last Punnett square. Big G, little g is a green pod. Genotypes, for your first mark, both of those green pods are big G, little g. And we know that we're undergoing meiosis to give us the gametes. Okay, we're splitting those G's apart, and we know that each of those green pods is going to give us a big G and a little g in either a sperm or an egg. Notice they haven't described which ones are males or females in this example. Doesn't really matter. We carry on down. And now we're in that Punnett square. These are where the big marks come from normally. So you can see that we've got big G in the top left box. We've got 2 heterozygous genotypes there. And we've got 1 homozygous recessive in the bottom right corner. If you've got all of this, so let me just highlight quickly. If you've got this bit and this bit right, that's 1 mark. And if you've managed to get the table filled in again, that is a 2nd mark. So well done if you got that right. But we're only 4/6 of the way there, 2/3 of the way there. The final mark comes from telling me the genotypes and the phenotypes. Now, I don't really like the way that they've written this statement at the bottom, a ratio of 3 plants with green to 1 plant with yellow. I would much prefer in an exam you spend the time and you go 1 out of 4 big G, and we know they're green. And we've got 2 out of 4 big G, and we know they're also green. And 1 out of 4 little g, and we know they are yellow. Okay, and then you can summarise that, saying 3/4 of them are green and 1/4 of them is yellow. It just shows in a little bit more detail that you know what genotypes give what phenotype. Okay, if you didn't get all 6 of those marks, pause me now, go back to the beginning, take a look at the rules again, and hopefully you should be able to succeed next time. If you're confident, however, let's move on. So for the next few slides that I'm going to change to slowly, I've just put some summary questions. If you are with the big blue book, I think it's page 423, and that's in the years 1 and 2 Oxford book. If not, I've put the pages on the next few slides, and I'll just click through them slowly and go through the answers as well with them on there. So you're going to be completing the summary questions on page 159 if you've got the year 2 book, or page 423 if you've got the double years book. Mark it using the back of the book, and then have a read through and make notes and complete all of the tasks on page 160 to 161. Okay, this is where we start looking at the mathematical side of genetics. We're looking at ratios and data and things like that. It's not too difficult. Don't panic if you're not a math magician yet. It's going to be fine. But yeah, your teacher will be on Teams for you to ask any questions if you're not sure. If you're in my class, these will also be uploaded to the OneNote page for lesson 7.2, so you don't have to worry about pausing the video from here on. Okay? So here's the first set of summary questions for page 159. Pause now. And here are your answers. I'm not sure whether you'll be able to zoom in on the video. If not, you might want to screenshot this page and zoom in and zoom out on your camera roll or something like that. And then here's the information from pages 160 to 161. Like I said there, it's on OneNote for my classes, just so you can access it a little bit easier. I'm sure Miss Goldie or whoever you've got will be able to help you as well. So spend some time reading through, making sure you're looking at the Mendelian ratio and understanding how you're going to calculate those ratios themselves. And once you've had a read through, you should be able to answer these summary questions. Can you please let me know if and when you finish them, and I will send you the answers myself if you're in my class. If not, Miss Goldie should be able to send them to you as well. Okay, thanks very much for today, guys. I hope you found it useful and informative, and I'll see you next time for dihybrid inheritance. Whoop whoop! Bye bye!